Hello everyone, Sanket here. Welcome to another video, another tutorial on my channel. Today we are going to discuss and look into what exactly is molecular dynamic simulations. Molecular dynamic simulation was first carried out in 1977 by J. Andrew McCammon. Okay, he performed the simulation on BPTI, or also called as bovine pancreatic trypsin inhibitor. The paper was published in Nature on 16th of June of 1977, and the PDB structure for it was later available after a few years in Protein Data Bank. The PDB ID for the structure, that is your trypsin structure, is 2CGA, so you can go and check it. So what exactly is molecular dynamic simulations? So molecular dynamic simulations is a simulation which helps us to analyze the movement of atoms or the molecules. Um, so each and every biomolecule has molecules and they have these atoms. When we are talking about MD simulation of a biological protein, this biological protein is there in our cells. Okay, and you know that all our cells have about 80 to 90% of water. Okay, so how do these biomolecules react, or they behave when they are kept in any particular solution? So it can be a water solution, or it can be some other liquid solution. Or how does a biomolecule react when they pass through a bilayer membrane? So this is all what you can analyze by performing simulations, or this is what we call as your molecular dynamic simulations. The atoms and the molecules which are there, okay, they interact for a fixed period of time. So this fixed period of time can be for particular nanoseconds, or it can be for a particular seconds. And this evolution, what we see, is called as a dynamic evolution. So when I'm saying the movement, I mean to say how these atoms or these molecules, they move when they interact with the different liquid molecules, as you say they are, or with the lipid bilayer molecules as they pass through the cell membrane. So all of this, I mean, by experimentally you can analyze, by looking at the peaks, or looking at the graphs, or looking at the color change, you can do that. But when you want to see something actual, how a protein is behaving, okay, that is where your bioinformatics comes into play. So you can say your MD simulation sort of shows a small short movie of how a protein behaves in a biomolecule solution. So your molecular dynamics, it follows a Newton's equation. Okay, and specifically it follows the second law of Newton's equation, where your force exerted is based on the mass and the acceleration what is there. So that means that if your mass is greater, the force which is applied to it will be greater. If your force is less, if the mass is less, the force applied to it will be less. This is the same concept, or the same funda of your when you're performing MD simulations for any biomolecule. So if your molecular mass of the protein, or the protein of any, or any other biomolecule for that matter, it can be a DNA or anything. So if your biomolecular mass is great, then the force which is applied for the atoms and the molecules to move will be great. If the molecular weight is small, then the force which is applied will be small. Okay, so yeah, so your MD simulations, what they do is they follow the second law of Newton, that is F equal to ma. Now, what are the steps of these molecular dynamics? All right, there are three crucial steps. Okay, there is creation of the initial step, then there is introduction to interaction potentials, and third is to predict how a particle will move. So this particle can be any of your biomolecule, as I stated earlier. So the first one, that is in your creation of the initial step, there are three substeps. One is to generate a topology file for the protein. Second is to add box and add the solvation of the system. Okay. Add box is you add a box around your protein. Solvation, that means you add water molecules to the system. All right. Or any liquid to the system. Third, in your initial step, is you add ions. So sometimes, you have biomolecules which do not have a particular charges to them, so there are some ions which are less. So in this case, you can add ions in order to bring equilibrium to the system. So these are the three steps in for your initial step. Then the fourth is introduction of interaction potentials, where you carry out the fourth step is where you carry out your energy minimization step. Okay. Energy minimization is nothing but where you bring down the overall energy of the biomolecule in order to make it very much stable. This energy minimization step, you can see if you're when you're performing docking. What exactly is docking? You can go to my other video of molecular docking using Autodoc Vina, where you can understand how energy minimization step is done. But in homology modeling also, you can carry out energy minimization. In docking, you carry out energy minimization. In MD simulations, you carry out energy minimization. And the last two steps is equilibration of the step and your final MD production run. Okay. So your equilibration of the system here is nothing but equilibration with constant pressure, with constant temperature and constant volume. So I repeat again, three important things: constant pressure, constant temperature and constant volume. Keeping these three things constant, we run the simulation for your biomolecule, what is there. So thank you.